Let's get right into it. Deep inside every single one of your cells, there's a master blueprint for life. It's called DNA, and it holds the instructions for, well, pretty much everything your body does. But how does a static code, tucked away and protected inside the nucleus, actually get out and build stuff? That's the amazing story we're going to explore today, the story of transcription.
I mean, really think about that for a second. Your DNA is like an incredibly rare, priceless book. It's locked away in the most secure library imaginable, the cell's nucleus. You can't just check it out and carry it over to the construction site, right? It's way too precious. So the cell has this huge logistical problem. How do you use the instructions without ever letting that master copy get damaged?
Well, the answer lies in this really fundamental principle that basically governs all of life. It's known as the central dogma of molecular biology.
Now, this simple little diagram was first thought up by Francis Crick, and it is the master plan. What it says is that information doesn't just go anywhere, it flows in one specific direction. It's a one-way street. The permanent storage, the DNA, is first transcribed into a temporary message, which is RNA. Then, that RNA message gets translated into the final product, a protein, which goes on to do all the actual work in the cell. Today, we are zeroing in on that critical first step, getting from DNA to RNA.
So, to solve that big problem of protecting the master blueprint, the cell just makes a disposable copy, a working copy. And that copy is made of a slightly different but absolutely perfectly suited molecule called RNA.
And just look at how perfectly RNA is designed for this job. You see, DNA is double-stranded, and it uses a deoxyribose sugar, all of which makes it incredibly stable. Perfect for long-term storage, right? RNA, on the other hand, it's single-stranded and uses a different sugar, ribose, which makes it way less stable. Now, that's not a bug. That is absolutely a feature. You want a temporary message to be just that, temporary. As soon as the job is done, it breaks down. This prevents the cell from just endlessly churning out a protein it doesn't need anymore. It is a brilliant system of controlled temporary communication.
And the hero of this whole process, the molecular machine that actually makes the RNA copy, is this amazing enzyme called RNA Polymerase. You can think of it like a highly skilled scribe. It goes into the library, finds the exact page it needs in that master blueprint, and then carefully copies the text onto a scroll that can be sent out of the nucleus.
So, the act of copying that genetic text is called transcription. And, while it can get super complex in the details, the basic process can really be broken down into 3 simple, elegant steps.
But hold on, before we dive into those steps, I have to share this with you. This is a description from a 1970 experiment by a scientist named Oster-Miller. He and his team looked through an electron microscope, and they saw something that looked just like tiny Christmas trees. A central fiber, the trunk, with all these branches coming off of it, covered in little granules. What in the world were they looking at?
This is what they saw. The trunk of the tree, that was a strand of DNA. And the branches, those were molecules of RNA being copied from it, getting longer and longer as those little granules, the RNA polymerase enzymes, moved down the gene. This was the first time in human history that we had actually seen a gene at work. I mean, what an absolutely incredible moment.
So, this whole process is highly organized. First up, in initiation, the RNA polymerase finds a specific start here sign on the DNA called a promoter. Then comes elongation, where it just chugs along the DNA strand, unzipping it and building a matching RNA copy as it goes. And finally, in termination, it hits a stop sign, lets go of the DNA, and releases that brand new RNA message. That Christmas tree image? It's the perfect picture of this in action. Dozens of polymerases all caught in the elongation phase at once. Find the start, copy the message, know exactly when to stop.
Okay, so that 3-step process is the basic version, kind of like what you'd see in bacteria. But in more complex organisms called eukaryotes, and that's anything with a cell nucleus, so plants, fungi, and us, that freshly copied RNA message isn't quite ready to go. It's more like a rough draft.
And this rough draft, it has to go through a few pretty intense rounds of editing and quality control before it's even allowed to leave the nucleus and be turned into a protein. Now, the reason for all this editing is that our genes are, well, they're interrupted. The actual coding parts, the important bits called exons, are broken up by these long stretches of non-coding sections called introns. Just like you see on the screen, it's like trying to read a sentence with a bunch of random junk words thrown right in the middle. To make any sense of it, you've got to cut out all that junk.
And this editing process has a name.
It's called RNA splicing. A molecular machine called a spliceosome comes in, snips out all those introns, and then stitches the exons together perfectly. It's what turns the junk blueprint junk must junk be junk read into a nice clean instruction, the blueprint must be read. And here's where it gets really clever. The cell can sometimes splice those exons together in different combinations. This is called alternative splicing, and it means that a single gene can actually create instructions for multiple different proteins. It's one of the main reasons complex organisms can do so much with a relatively small number of genes.
But wait, the cell's not done yet. After splicing, the edited message gets 2 more modifications for protection. As you can see, it gets a cap and a tail. A special 5' cap is added to the front end of the RNA. It acts kind of like a helmet, protecting the message and also helping it dock with the protein-making machinery later on. Then, at the other end, the 3' end, a long poly-A tail is added, which basically increases its lifespan. Only after it's been spliced, capped, and tailed is the RNA message finally considered mature and ready for its big journey.
Okay, so let's zoom back out. Why does this tiny, super intricate process matter so much?
Well, for one, because it is happening at an almost unbelievable speed. That RNA polymerase scribe, it's not taking its time. It's adding somewhere between 30 to 50 new letters, or nucleotides, to that growing RNA chain every single second.
And that right there is the crucial takeaway. This entire dance, from finding the right gene to transcribing a temporary copy to editing and protecting that copy, is happening constantly in almost every single cell in your body right this second. It is the fundamental process that turns the static information locked away in your genes into the dynamic living reality of you.
And that leaves us with one final, fascinating question to ponder. A skin cell and a brain cell in your body have the exact same DNA blueprint, the same master instruction book. So, how does a skin cell know to only read the skin cell chapters, while a brain cell only reads the brain cell chapters? Well, that is the secret of gene regulation, and it's a story for another time.